we're gonna get started. Lance is here from Omega, so I'll let him introduce himself and kick it off.
From Omega. Yeah, should I launch into it?
Yeah, go for it.
Well, good thing is we have some samples today, but I'm going to be talking about some yeast strains that we've developed that we call thiolized yeast. So they're just yeast that can put out these thiol compounds that have a lot of very tropical aromas to them. And it's doing this from precursors that have always been naturally present in hops and malt. So it's just a way to get more aroma out of the ingredients we're already using. So we call these thiolized yeasts. Should it be advancing?
So, yeah. There we go. Just click down.
Yeah. Okay. But before I launch into some science, a little bit of background about myself. I have a bachelor's in microbiology from the University of Illinois in Urbana-Champaign. That's where I started homebrewing. We had a university sanctioned club at that time. The club still exists, but it's no longer university sanctioned. But I'm glad it was at the time. Otherwise, I may not have obviously had a big impact on my life. But from there, I moved down to University of Texas in Houston and also started working with yeast there, but it was more in like the human health context. So we studied molecular chaperones, which are compounds enzymes that we have in our own cells, but we studied in yeast, which is a lot easier to do. But I took a detour from there and went to law school, and there was a plan, but I ended up being a patent attorney and did biotech patent law. So you have to have both a technical degree and a law degree, because you're essentially acting as the go-between between the inventor and the patent office. So you translate their invention into a legal language, into a patent application. So you have to understand both sides of things, but ended up not particularly enjoying being a patent attorney. It's fun to be, involved in cutting-edge science and all that, but it's also pretty dull. But ended up having a very chance conversation with one of my colleagues at the firm who was starting a brewery in Chicago. If any of you have heard of 1090 Brewing in the north suburbs of Chicago. So one of the co-founders was one of my colleagues at the firm. And we just were talking at a firm event one time, and that conversation about where he was getting his yeast just spawned this idea. So it wasn't on my radar before then, but went home that night and started planning opening lab. So this was in 2013 that we launched, along with, so I have a partner who was also an attorney at the firm. He's an electrical engineer, whereas I was more on the biotech side. So he takes care of all the business stuff, and I do the production side. So as I mentioned, all of our production and research happens in Chicago, but we do have an office in St. Louis too. That's where our business partner's from. So we moved back down there, and that's where we have business and marketing activities take place in St. Louis. All production and research is in Chicago. There's me, clean shaven. And I don't normally, so it's become somewhat of an annual tradition to shave my beard like a weirdo like this, because I've dressed as Lemmy from Motorhead for the last few years. So that is why I have this. So then I just run with it for a while because I look like a weirdo. Anyway, so overview of what I'm going to be talking about today. Biotransformation generally. It's a word that brewers throw around a lot, and I'm going to give you a little bit of science as to what that means, explain exactly what thiols are, and talk about thiolized yeast and what we mean by thiolized yeast and what they're doing and how we engineer them, and then talk about where the fuel for these thiolized enzymes are coming from, malt, hops, and wine grape derived products, and then give you some tips on maximizing it when you want to maximize it, minimize it when you want to minimize it, and also some of the additional research we're doing. So biotransformation generally the transformation of chemical compounds within the living system. So just taking things that are present naturally in our hops or our malt, and through the activity of yeast, converting them to fruity aromas that we find pleasing. So why do you want to care about this? It's because you can push, new aromas and flavors with the ingredients you're already using. Also just for higher quality beer and just understanding how all the flavors in your beer interact, which is one of the fun parts of brewing, is kind of discovering these things. And then there's also a sustainability argument there, because we can get more aroma, more punch out of the ingredients we're already using. If we can use yeast to kind of pull out more aroma and more flavor from the ingredients that we already have. So there are a lot of different compounds that make hops smell like what they smell like. We're gonna be focusing on a few where yeast kind of interplays with them. The most well-studied is probably the monoterpene alcohols. So a great example of that is geraniol. Yeast can take geraniol, which smells like more of a floral, flowery aroma, and it can convert it to beta-citranolol, which is more citrusy. So that's well established that yeast can do this. It's variable, but from yeast strain to yeast strain, not all strains do this equally. There's another very not well-known pathway is hop esters. So hops have some amount of esters, and yeast can actually break down those esters, so kind of minimize the effect of the esters that the hops are contributing. But again, that's not terribly well studied. And then what I'm going to be talking about tonight are thiols, which are a class of sulfur-containing compounds that are extremely aromatic in very, very, very small quantities. I have some nice pictures to give you a sense of how aroma active these things are. So what I'm going to be talking about tonight then in particular are precursor versions of these thiols that are normally present in malt and hops, and in the form that they're present, they're not aromatic. So they're there, but they're kind of inert. And if we give them the right yeast that has the right enzymatic activity, we can convert those thiols from inert compounds to aromatic compounds through the magic of biotransformation. So thiols are found in a lot of tropical fruits. That's what gives them their aromas. We're going to be talking a lot about one particular thiol called 3SH. And it has aromas of grapefruit and passion fruit. And 3SHA, which is a very similar, it's a acetylated version of it. So it's very similar to 3SH, but it has a lower aroma threshold and smells very much like passion fruit. So the threshold for these is, 4-MMP is one you'd probably also be familiar with if you used citra hops, and if you've ever gotten citra hops that are what we call catty, so they smell like cat piss. That is 4-MMP. So at very, very small amounts, it gives you box tree blackcurrant aromas, but when you get to a certain level, it starts to smell more like cat pee. But that's at 1.5 parts per trillion. So, I mean, absurdly small amounts. And I will illustrate this here in one of these future slides. 3SH takes a little more. It's in the order of 60 parts per trillion until you can smell it. 3SHA is only 4 parts per trillion. Some of these other thiols are provided by hops too, but don't seem to have any sort of yeast biotransformation component. They're just there in these kind of free forms that we can smell. If you've ever had a New Zealand Sauvignon Blanc, and if you haven't, I would recommend you do, because what gives those their characteristic aroma are these thiols. You might, if you've had them, and some of these, maybe the Star Party beers we're going to try, you might recognize some of those aromas from New Zealand Sauvignon Blanc, and that's because those are very, very important compounds in those wines that make them smell like they do. Because these precursors are actually present naturally in some wine grapes too, especially in New Zealand wine grapes, and also New Zealand hops. So if you've ever had Nelson Sauvin, it's thiols that give them their kind of unique tropical aromas and wine-like aromas.
Does that come from soils, stuff like that that are down there?
Not particularly well known. Probably that's part of it. Interestingly, it's also how they harvest it. So a lot of places are very careful about picking grapes carefully so they don't damage them before they crush them. But in New Zealand, everything's mechanized, so there's a lot of damage while they harvest them. And these style precursors are present in the grape skins. So, and they've shown this, that a lot of it happens when they harvest it and the skins get damaged, it leaches into the juice and then it's available to the yeast to convert to that. So some of it even comes down to just how they're harvested. They seem to have a lot, but mechanization of it has also led to it, which is kind of weird. Wouldn't have thought just how you harvest can impact tropical arose, but that's one of the things.
So it's only in the skins?
Seems to be.
Okay.
So this gives you a sense of how aroma active they are, how little you need. So if we're pretending our beer is an Olympic-sized swimming pool, if we look at some of these other compounds, for example, esters that give you fruity aromas, if you had 5 beers to 21 barrels worth of beer dumped into that swimming pool, that's how much ester or aldehyde you need to be able to smell. Diacetyl is a little more potent, and you'd need 2.5 milliliters to 5 beers worth of diacetyl dumped into that swimming pool before we can smell it. So again, even that, it's not very much. But then when we get to thiols, we're talking about drops. So just a drop of water into an Olympic-sized swimming pool is enough for us to smell a thiol. So humans are remarkably sensitive instruments for smelling thiols and sulfur compounds in general. We're generally very, very sensitive to sulfur compounds. Some we like, some we don't. A lot of like onion flavors, garlic, all those sorts of things are thiol aromas. It takes very, very little for us to be able to smell them. So what is it that yeast needs to do to these precursor forms that are inert to be able to smell them? So this is, on the left is what they look like when they're in that precursor form. So they have, in this case, it's an amino acid that's linked to the thiol. So the thiol's on the bottom, the amino acid's on the top, and there's an enzymatic activity called beta-lyase activity that can just clip that cysteine, that amino acid, off of it, and it releases the free thiol that we can now smell. So on the left, we can't smell it. On the right, we can now smell it. So it's this one critical enzymatic activity that we need to be able to. So there are lots of versions of these precursors there that range in complexity. So the cysteine-bound version of the precursor is the most simple and the one that the beta-lyase enzyme in the yeast can act on. But there are more complicated versions of them that are bound to things like glutathione, a larger molecule that requires some processing to get to the version that yeast can act on. So I'd already mentioned New Zealand Sauvignon Blanc. That's really kind of where most of the research has occurred with thiols and how yeast can convert them. And in that research, they found that there's an enzyme called IRC7 native to yeast that can perform this activity. So it's the enzyme that's thought to be critical to get it making New Zealand Sauvignon Blanc smell like it does. And it functions on that cysteine-bound version. So yeast can bring in the cysteine-bound version of the precursor. The enzyme IRC7 clips off the cysteine, releases the thiol, and now we can smell it. It was also found in wine studies that there are a lot of strains that have inactive versions of it, but also that its expression, so the yeast making that enzyme, is regulated by the amount of nitrogen present in the must. And if you make wine, you probably know that wine has grape must has much less nitrogen than wort. So this is why, and this enzyme is regulated by the amount of nitrogen present. So if there's a lot of nitrogen present, it doesn't make the enzyme. You don't get the thiol release. It's thought that the enzyme is actually used to scavenge nitrogen from the environment. And in wort, there's always abundant nitrogen levels, so this enzyme is never made. So this is why you've never really heard of this until now, because brewing yeast might have this enzyme, but it's never ever made in brewing yeast because we always have abundant nitrogen available in our wort. A mind of its own. So we had to figure out a way to get this enzyme to be active. So the first thing we tried was just fermenting wort with wine strains that are marketed to be high thiol producers. So we did that, and we really couldn't smell anything different. I mean, it smelled kind of winey, but they were wine yeast. Nothing that we would describe as being very tropical. So we also crossed, bred or mated wine strains with beer strains to see if we could get anything that way. So again, tried a couple of strains that are marketed for being high thiol producers, and with our hazy IPA strain, and even with that, we did not get any amount of thiols that we could perceive. So even a wine yeast that makes a lot of thiols in the wine context, it's that nitrogen regulation that kind of stymies it when you put it in wort. So there's tons of nitrogen around. That's how the gene's just not made. We don't get thiol release in these strains. And even then, there's also, even if it has a functional version of this gene, sometimes they're also broken. So they have a mutated version of it. So we're kind of out of luck with most brewing strains. So the next approach we took was, so I mentioned that it's controlled by nitrogen. So we wanted to change what's called the promoter, which is just a region of DNA in front of a gene that tells the gene when it should be turned on. And again, the native version tells it not to be turned on when there's a lot of nitrogen. So we put a promoter in front of it that's just on all the time. So regardless of what the nitrogen content of the medium is, it's going to make this enzyme. So we did this in both our 004, which is essentially the Chico strain, and our 011, which is our hazy IPA strain. And on the left is the amount of 3SH which is released or in the wort when we have not done anything with the strain. And on the right is a modified version of the strain where we replace the promoter in front of the native IRC7 gene in those strains. And what we saw and smelled when we tried the Chico version, the 004, is we got a spike in 3SH. So all of a sudden we could ferment wort, we could smell tropical aromas just coming out of that wort. So this tells us that the enzyme inside Chico is actually, it functions, it's working, but it's not made. So if we can artificially get it to be made, we can smell thiols. But British 5 has that other problem that I was talking about, where it has a broken version of the gene. So it has the gene, but it's got a mutation in a critical site that means it can't do this conversion. So even though we've relieved that negative regulation by putting this alternative promoter in it, we still don't get release from the British 5 version. But we really wanted a British 5 version of the thiol capability, because that is our best-selling strain. It's the strain that lots of people use to make hazy IPAs. So we wanted to make this popular strain better, but we weren't going to be able to rely on the native version of that gene. So what we needed to do was take that functional version from Chico and put it into British 5. And the way we do that is using CRISPR-Cas9. So if you want this won the Nobel Prize, no, not us, but CRISPR won the Nobel Prize last year. And what it essentially is a tool for breaking DNA. So you can kind of program this enzyme to cut DNA exactly where you want it to cut it. So we've historically used with our engineered strains the FDC1 gene, which is what's responsible for making phenolic aromas in Belgian yeast. But in British 5 and Chico and all these strains that we regularly use, that gene is already broken anyway. So we use that as our site for incorporating DNA. So this enzyme, this Cas9, CRISPR-Cas9, we can program to cut in the FDC1 site, and we supply DNA to that cell that has this alternative promoter linked to the IRC7 gene that we know works, and put that in the cell. And where it's broken, the cell repairs that break, because a double-stranded DNA break in the yeast cell is an existential crisis. It will die if it doesn't fix it. So it loops in this DNA, and now we have a yeast strain that is making IRC7 and releasing thiols. So this is, we're very open about how we do things. This is how we do it. We're just, in this case, kind of rejiggering yeast DNA. We've taken 2 pieces of yeast DNA from a different strain and plugged it into this strain.
And
So-
this was Cosmic Punch,
Yes, this
Which
is,
is
this is
the-
yeah,
3 sheeps, and
And-
it's Cosmic Punch. So-
and this is what you're.
Wait,
so,
you're
yeah.
drinking already?
Yep.
I see. Dash your glasses, people.
Not that,
Give it
yeah, I
a
try to
try.
slam everything when it's done.
Lance, I have a question.
Club,
Yeah.
give me a cup.
I mean, you know, I read the book by Walter Isaacson about the discovery of the CRISPR process. How easy is it to do that?
We will
I
make
mean,
it
how do you
happen.
technology to do it? It seems like we get great things from the lab.
Right, right.
What's going on?
So CRISPR, I mean, it takes some know-how, but once you have kind of the DNA constructs in place, I mean, we can order like a piece of DNA we're wanting to look at. There are services that construct that for you. You just send them and make it, send it. And then, so we can just put that all in yeast cell.
So I mean, you get that from some other lab?
From IDT.
Oh, you got it?
Like,
Oh,
so
sorry.
DNA, send them the string that you
So actually,
want.
nine.
I know you've got a PhD in microbiology, so I mean, it's not, it seems extraordinary that you've done this. Your yeast.
Yeah, well, and we're also lucky that yeast is very amenable to these sorts of things. So it readily takes up DNA. It has robust, what we call homologous recombination, which is what we're taking advantage of here.
Is that drinking
Um,
hot drinks? Yeah.
For the piece that we're wanting to insert, we surround it with the DNA that looks like the DNA where we're cutting it. So that's what causes it to leave it in. And it just does this very, very readily. So we can kind of just trick
Like a
it.
chemical reaction.
Yeah, it's totally chemical. It's enzymatic,
Yeah.
chemical, yeah. So it's just giving them the right collection of chemicals to take up the DNA. So this is what Cosmic Punch is. So it's the IRC7 gene from Chico with that TDH3, the constitutive always on promoter, put into British 5. So a native yeast gene that normally isn't on in when we're making beer, that's now on, that releases these compounds.
So if someone makes a beer with this, they have to call it Jurassic Park.
I don't know if they have to, I guess they can.
I was just in the 90s.
So we've, since then, we've kind of moved to a different version of a thiol releasing enzyme, and that is the PAT-B gene, the PAT-B enzyme that comes from the bacteria S. hominis. So this bacteria is actually native to our skin. And what it does on our skin is actually, it takes, so we secrete a precursor, much like these precursors that are present in malt, but it's slightly different. So it has an additional methyl group on it. So the bacteria takes up this, and it's doing it for the same reason. It's trying to scavenge nitrogen from these things that we're secreting from our glands. So this bacteria can take it up and release this thiol called 3M3SH. So it's not 3SH, but it looks very, very similar to it. But that aroma, or when that's released, that thiol is what gives us body odor. So it's an extremely similar compound, but thankfully, you know, malt and hops don't have 3M3SH. Otherwise, it would smell like body odor. But the precursor is so similar that this enzyme can act on, but it does it much, much more potently. So I'll give you an example of some of the levels. So Star Party, which is one of the other ones we're going to try, is the Chico strain that has this bacterial version of the enzyme called PAT-B that's on all the time that can release thiols, and it just does it at a much higher rate.
I'm sorry, I might have missed something, but so the way that you CRISPR these together, you don't have to reduce the nitrogen amount in the wort in order to achieve that same flavor.
Yeah, it's just, yeah, so the promoter is what tells the enzyme when it should be made. And we've just given it a promoter that, it's what we call a housekeeping promoter. So it's normally linked to a gene that's needed all the time. So it's just tricking the cell into making this enzyme all the time. Even though it doesn't really need it, because it doesn't need to scavenge nitrogen. We want it for this thiol releasing capability. So we kind of have two classes of these thiolized yeasts. Cosmic Punch relies on IRC7, which is a native yeast enzyme, and Star Party, Heliogazer, and Lunar Crush rely on the PAT-B. So as far as like potency, Star Party, Heliogazer, Lunar Crush are very, very much more potent than IRC7 at releasing these thiols. So where do these precursors come from that the enzymes are acting on? I think one of the most interesting things about this is that the most abundant source is malt. So we think of hops as always being the source of the aromas that we get in beer, but malt is just chock full of these precursors. Hops also have them, and hops have some free thiols and bound thiols, but they have less. So both of these things are sources of kind of fuel for these enzymes. And one of the interesting things is it varies. The amount of precursor varies in the type of malt you're using, or even the type of grain. So this is giving you an example of the precursor. So this isn't released thiol. This is the precursor and all those different versions of the precursor I showed you in different grains. So we have two row on the left, Munich malt, Vienna malt. And you can see the levels vary depending on which malt we're using. And we found that at least the ones we've tested, the white wheat and oats, have none. So what's that? I know. Yeah, this is all we know at this point. That it's in barley, but it doesn't seem to be in wheat or oats. But I also said we haven't exactly, this is white wheat. We haven't tested red wheat yet. We haven't tested rye. We might find it in other places, but at this point, we know it's in barley. It gets destroyed when you kiln it more heavily, so you won't find precursors in like chocolate malts or things that are more heavily kilned. It's more kilned than the two row, but this also might have been like the base malt that they used was higher, because there's one study that was done by a French lab that showed just different base malts have different levels. So whatever base malt they used for that Vienna might have been higher than the two row that we were using the rest of this. Unfortunately, at this point, there hasn't really been an exhaustive, and this is not something that maltsters include in their normal analysis. It's something we've been bugging them to do because now it's more relevant. What's that?
How difficult is it to test for this precursor?
Well, we can't. So every graph you're going to see here, every bar graph, is a result of a sample that we sent to France. So there's a lab that was kind of instrumental in studying this in wine that does really, really good at quantifying these things. So each measurement you see in here is essentially $300, which is why we haven't done the exhaustive analysis, because it's extremely expensive.
Deal. Understandable.
But I think it's going to be worthwhile knowing this sort of thing, or getting this capability domestically would be good too. So the way that they do it is they can create radioactive labeled versions of these compounds, and measure those, and then have a relative amount compared to the sample you sent them. So it allows them to quantify it very, very precisely. There are labs over here that can qualitatively measure it. They can say, yes, it's there or not, but they can't reliably tell you how much is there. So when we want to know how much is there, we send it to France.
So it's a chemical thing rather than like is it a spectrometer they used in order to
Yeah,
find
it's a GCMS with a particular type of column, but and those things exist stateside. It's just that nobody else is making these deuterized standards that they use to really quantify. So you do find some labs that are detecting these, measuring them, but not quantitative, not in a way we could. And we're trying to get relative differences between malt varieties and between yeast output. We want to know. We want to be able to say with confidence what the quantitative differences between these samples. Not just we know it's there. We want to know how much is there and what conditions push it up and what conditions push it down. the only lab we found is that one in France. We sent it to a separate lab in France that gave us very, very different numbers. So we've only found that one lab that we fully trust to do this.
You want to make your own?
We have a GCMS, and we were going to get some I mean, we don't really have a chemist to be able to do this, but we were going to get standards from them to see if we could do it in house, and then we could potentially just order standards from them and do it. So we haven't gotten it. Be nice, because this is extremely expensive to do this, but we've learned a lot. So this is well, and then, I mean, if we compare this, which is the vial that's released by the thiolized yeast, it doesn't even always add up to the precursor. So there's something else going on there too that's impacting the amount of thiol that the yeast puts out. So it seems it gets like complication compounded on the complication the more we look into this sort of thing. But to remind you of the aroma threshold, 60 parts per trillion for 3SH, 4 parts per trillion 3SHA, and then what we get in the non-modified 011. Compare that to, this is now heliogator. So the other numbers that we saw before were cosmic punch, were more on the order of like 300 to 500 parts per trillion. So you can see how much more potent this PAT-V version is. So in just 2 row, we get, we're in the, 10,000 parts per trillion. So it's really, really potent, like 100-fold above the aroma threshold for it. And there's no hops here. This is just small.
You get so much of the precursor that it doesn't, that it's too much in the precursors, but it doesn't make the negative reflect to it, but it's too much precursors.
That's somewhat in the eye of the beholder. I mean, there are some beers we've made that are weird. And these are polarizing compounds. So if any of you start to play with these sorts of things and do like a blonde ale, for example, that does where all you're smelling is the thiol, some people are like, that's too much.
So do you give any guidance on how much that we should be adding to? It sounds like it doesn't take nearly as much hop in order to get these types of aromas.
Well, but what complicates it is when you do, which I think we'll smell here, because you did a IPA with. So what was your total hopping level?
Yeah, so we can do that one next. So I use Star Party.
So we'll be putting out on the order of this much. It's the same enzyme that is Ecobacter.
Rock with just like 2.5 ounces, about a little over 2.5 ounces.
Right.
But I
Feels like too much.
did a mash hop with 4 ounces of Cascade and Whirlpool 4 ounces of Idaho 7.
I always have to convert this to, we always
Grams.
pounds per barrel.
Oh, sure.
So where are we at? Half a pound per barrel.
So kind of that,
I'll read it to you.
the question that's in my mind, which I'm having a problem with. We're worried about all the acid levels. We're worried about co-cumulons. Is this just how do we tie in the fields to that?
It's going to be. We don't have all the answers because we don't know the precursor status of the. But also, I think what we'll kind of figure out here is that how much you're hopping vastly affects how you're perceiving this too. So if you do an extremely low hop here, it's dominant. It's pungent. You can't notice anything else. All you smell is like weird tropical aromas. But you put in 1 pound or 2 pounds per barrel of hops, even though we can objectively measure that same amount, it does not come across anywhere near that, which is very weird. So there's like masking going on. Right. So it's, it very much depends on the beer you're brewing. So
Yeah.
some of it's going to be trial and error, just with the materials you have in your possession. We can't try everything. So there's, an element of exploration with this. We don't know. And it might not be good. I mean, there might be contexts where it's not good. And we're still trying to figure out like which hops and meld with better.
Probably looking for feedback all the time, right?
Yeah, I mean, and we get some that, some people that like, I don't like it. Not offended by that.
This is the Star Party.
Star Party.
I made this one.
Nice, Andy, nice. Nice shirt, by the way.
I hope so too. I hope
It's coming across more prominent
hope
here.
it does it well.
I love that shirt. I think it's amazing.
Hey, quick question. So, one of my current practices is dry hopping at like the high Charleston to try and get more aroma into it. How does that affect it, or is it worth it, or something?
There's an order.
Or am I moving ahead here?
Well, I'm not sure if I have, we've done,
Right?
I have slides, I'm not sure if they're in this presentation. So what we have found, I know I've got some, we can skip this, these precursors are boring. I think I will get there.
Okay.
This slide's just showing you that much like the malt context, there's variable amounts of these precursors present in different hops. And interestingly, some of the hops that are most abundant in these precursors are like, European continental hops, like Sobs is loaded with precursors. So, there are hops that you would not think of as being as sources for tropical aromas that now can be thought of as hops that we can get tropical aromas out of by using them in certain ways at certain times. But this is it. I mean, the research on like the amount of thiols present and precursors present in hops, this is it. There really hasn't been a robust study on that either to say, how much does it matter what field it came from, what year it came from, where it was ground, how it was processed. We don't know any of this stuff yet.
But as far as a time once you add it, have you figured out?
So yes, that, and I will have some slides. So this brings us to mash up.
Oh, sure.
So one of the ideas that we had, based on the kind of collection of precursors present in hops, a lot of it is that glutathione version that the yeast can't act on. So we thought, what would happen if we put hops in the mash, which is full of proteases and stuff that could process these more complicated versions of the precursors, and then knowing that they're soluble and non-volatile, they should get extracted into wort and make it all the way through the brewing process until they're available to the yeast. So these aren't going to be boiling off during the boil. So this was the thought behind mash hopping and why mash hopping might lead to more precursors available to the yeast to release more thiols. And we found that we did get more thiol release with mash hops. So when we add you see no hops at all. With Cosmic Punch, we get 323 parts per trillion release of thiols. When we put hops in the mash, I think this is, 1 pound per barrel. Chinook on the left and Montewake on the right. We get an increase to 388 parts per trillion. So putting hops in the mash released more thiols, made more precursor available to the yeast, and we got more thiol release. What we saw that we did not expect, and we've seen repeatedly at this point, is if we added those hops to the whirlpool, we saw a decrease in the amount of thiol output. We still don't know why, because you would think that even if it wasn't contributing to the pool of precursor, that you'd at least be at the same level as malt alone, but in fact, we see a decrease. So we don't know why. There's something in the plant material that's oxidizing the free thiols, that's dragging it out of solution. We don't know. But when you use them to mash, we don't see that effect. It actually does seem to be adding to the precursor pool available to yeast. But if we add those same hops to the whirlpool, we see a difference. Yeah.
So the whirlpool bar is with no hops in the mash, just the whirlpool.
And what was that whirlpool temperature? 180?
Yeah.
You're basically saying don't whirlpool anymore.
If, well, but I mean,
For if you want, if you,
So that's what they.
if your goal is maximizing tiles.
for New England, which probably would be, right?
But you know, if you don't do that, you're not going to be getting those things that are normally contributed by whirlpool. I mean, that's when you're solubilizing a lot of monotributing alcohol that you do want. So, but I would also say that 195 parts per trillion is still above threshold. I mean, you're still getting, so even if you do that, you still have thios that are well above threshold, well above anything that you have in a non-thiolized. So it's, yes, it's less than if you have malt alone, it's less than if you do mash hop, but it's still potent. And we did a repeat of this recently. That's on the right. Matahueca saw the exact same pattern. We do this on our wort brew house that we use for making wort for the yeast. So this is a nice automated system that we can tightly control, and we got very similar results in the 2 repeats. Get these questions a lot, which is why I now bring it up. So there are things to consider when you're mash hopping. Mash hopping does contribute to bitterness. And the kind of the rule of thumb there is you get 30% of the IBU levels that you get if you use that same amount for a full boil. So to use very simple numbers, if your calculator told you that adding X amount of hops to the boil for the full 60 minutes would give you 10 IBUs, adding that same amount to the mash would give you 3 IBUs. That's kind of the math you've used to determine what your IBU contribution is going to be. And so the rates, again, very much depending on the hops you're using and the alpha levels in those hops, is between half a pound and 2 pounds per barrel. So you can go to 2 pounds per barrel of sods, which tends to be very low alpha. Also happens to be very chock full of the precursor, and you can add a lot of precursor to it without affecting bitterness too much. So we've gone to 2 pounds per barrel and gotten like 18 IBUs, I think, from that. But if you use 2 pounds per barrel Chinook, or Cascade or something that's higher, you're going to get a substantial amount of bitterness from that. So keep that in mind when you're formulating your recipes. Also, all the old rules apply. So if you're adding citra to the mash, you're not going to get citra aromas out at the end, because all of those aromatics that we're used to from citra are going to be lost because you're going, it's at high temperatures for a long time, you're doing the boil. All that stuff's lost after extensive boiling. So don't use really, really expensive, nice aroma hops in the mash. So like I said, like Sots works well, although it's probably not something you're going to want to use because apparently the Sots crop in Europe was shit this year. They got like half as much as they got last year. So Sots is probably going to go through the roof. But it happens to be good for thiol precursors. But don't use citra, don't use mosaic, things like that in the mash, because you're going to lose all the magic that you're used to from citra and mosaic.
So would you suggest something with a high alpha acid level, small amounts, or would you play any part into that too?
Don't know about that, but I mean, that's right. Again, I think how much you use is going to depend on your goals for the recipe. And how much you're willing to put in the mash and have contributed to IBUs. And if you don't want much contribution of IBUs and you want as much thiol as you get, use Sots. Because if you need more of this, less. If you just want a little nudge of it, Cascade is okay, but use less than bittering. Work with it. And so again, something else I want to get, mash hopping is never necessary. There's loads of precursor from the malt itself. So you don't have to use hops in the mash to get precursor available to use. So there's already lots of precursor available if you have that malt. But depending on your recipe, it's high in adjuncts, high in wheat, high in corn, things that don't have a lot of precursor, then that might be a time you want to use mash hops, because then you might want to be able to reduce the amount of precursor available. Because those grains are we repeated the same thing with Heliogazer. So we saw the same overall trend, but again, remember that the strain is far more potent. So we saw an increase in thiol release when we mash hop. We saw a decrease when we added the same hops to the whirlpool. But even then, we're at 5,500 parts per trillion, so massively above threshold. So yes, it's decreasing it, but you're massively above threshold. So you know, don't be afraid to add hops to the world. Yes, it's decreasing the amount of vials it would otherwise put out, but you're still above. So kind of knowing all this research about Sauvignon Blanc juice being a source of thiol precursors, we tried an experiment where we dosed wort with 5% Sauvignon Blanc juice and saw a 20% increase in the amount of 3SH output, which is again showing you that Sauvignon Blanc juice happens to be a good source of thiol precursors. So also gives you an opportunity to make some kind of unique Eno beers, wine beer hybrids, and get some unique flavors out of it because of these yeasts. Around the time we were releasing Cosmic Punch, we saw this a couple breweries using this product called Phantasm, which sounded interesting because it was derived from the skins of Sauvignon Blanc grapes, which we knew from the research to be high in precursors. And so this Kiwi was selling these to brewers ostensibly to increase the amount of vials in the beer. But we realized that that probably wasn't going to be useful without thiolized yeast because, again, like natural yeast don't have the capability. You can feed them all the precursor you want. They're not going to release the thiol because they're not making the enzyme. So we got a hold of some of this and tried it with our engineered strains. And we do see a little bit of bump. So there might be a little bit of free thiol in the Phantasm because even in the British 5, we get a bump to 87 parts per trillion. But we go from 417 parts per trillion with malt alone to 867. So a twofold increase in thiol output by using 2 pounds per barrel Phantasm. So Sauvignon Blanc, great. Skins add a huge amount of thiol precursor to the malt and make it available to these yeasts that release thiols.
What's a Phantasm? You have to use the thiolized yeast. So
I don't tell Joss I said that, but that's I mean, that's
So yes, but not yes.
yeah.
Do you believe in
I think this is just well timed that he came out with this around the time that yeast came out that could make full advantage of it. But yes, in my opinion, it's you want to use a thiolized yeast with this to get the
Maximum potential.
Yeah. So just to kind of show you the difference and maybe the different utilization you can make of these strains. So to review, Cosmic Punch is based on our British 5 strain, so a hazy IPA strain. Utilizes overexpression of IRC7, which is a native yeast enzyme, and gets thiol output around 10 times the sensory threshold. So it's not nearly as dominant as the newer version. So you can use this in an existing any IPA recipe to just kind of give it a little more, but it's not going to be in your face and dominant. If you want in your face and dominant, use the newer generation of strains like Star Party, which is based on the PAC-B enzyme that gets you in the order of 300 times the sensory threshold. So you can use it to make, very weird beers. If you use it in a blonde that has no other hops, you smell like something you haven't smelled before. But also do a West Coast that's got aromas you haven't seen in the West Coast before. And this on the bottom is illustrated essentially how much thiol that strain is adding to the pool. So Cosmic Punch is adding on the order of microliters worth of thiol to the pool, whereas Star Party is dumping a shot glass of thiol into that pool. But again, it's still not that much, right? Like especially if you taste a blonde ale made with Star Party, I mean, you smell nothing but thiols. But that's realistically what you're adding to that beer. A shot glass into an Olympic-sized swimming pool. Very, very small amount, but a huge effect.
So we have another beer Star Party from Alex from
Wait.
Manitowoc
Say that
Club.
again?
It's another stop. Right?
No, another beer brewed with Star Party from Alex up in Manitowoc. The recipe's on the growler if anyone wants to take a look at it.
Was that the cute flower?
No.
That was also.
What was in that?
That was Star Party.
Oh, okay. Got it.
Wax.
Yeah, I think.
And Alex actually did 3 recipes with that. So bad. I don't work with it.
One was hydro.
Oh, sorry, one was the carpet. Sorry.
It's just like.
No, they have this, they had the 3 liquids. But it doesn't.
That was not supposed to be available in the US.
Well, he's been getting other stuff.
Yeah.
It's starting to style. It's a lot of stuff.
No, it's the neat one.
I don't know, he gets it though.
Yeah.
He gets it.
Black market.
I like Scarnets, but I would also note that it's based on
I served you last time, I'm not serving.
natural, it's got the natural regulation to it, so I'm not.
He didn't say Star Party because it's hydro, is what his.
It was literally set up.
That's right.
I think it's mostly his control.
Yeah, we're out on the same page. Some of the other style strains you
Under pressure, is there any difference?
I don't have any data. I would expect it to trap more, so it might make it, but these strains can also make a little more sulfur compounds too, so you might trap some things you don't want to trap, but you might want to off gas. So it's not something we probably need to experiment with. But my guess is it would trap more, which might be good.
Is it, I would think, is it more of a homebrew thing to implement under pressure compared to large brewery?
I think a lot of breweries use it at the end, funding to trap, to help with carbonation.
Okay.
Because it's too costly.
Yeah, and sometimes can't.
We got a question from, so Alex asked a question, who brewed the beer that's going around now, and he asked if you have done any tests on spent hops in the mash.
No.
Spent dry hops.
I mean, we've done experiments that use like the material that's left over after they.
That's like all the matter.
Yeah, and that contributes some. So it seems to kind of stay back.
Want some of this?
I smell something. I like your hair better. So it is, they last night or whatever said the best uses for the Star Party were like West Coast.
Yeah.
But this is a neat player,
No,
right?
I mean, which one?
Alex is.
Yeah, I think it's more based on New England.
So like, out of the gate, it's not like the best use for that, right?
So he was trying to do like a experimentation to see what things contribute to the same recipe.
Okay,
So he brewed it 3 times with Hydra, from Escarpment, Cosmic Punch, and Star Party.
okay.
Yeah.
Where are the other 2 to try? Just kidding, I'm just kidding.
What was it?
How should we
What was his, did he log his results? What was the?
Yeah, I guess, I don't know if he's still on, but Alex, if you're.
Yeah,
I'm here.
Okay, yeah, I guess, ask him what his favorite was of the 3 yeast. And it better not be Hydra.
Oh.
Well, I mean, like I said, these can be polarizing.
Yeah.
It's not something you're necessarily used to in this context. So I think. So
I like that.
yeah, it's gonna depend on what your goals are, but if your goals are really just to maximize the file output, things you could do are mash hopping.
I won't tell you what he said.
So not server.
It's all greens.
Yes.
What Guinness yet? It's
Like the original low calorie.
So we've had some theories that things like cryo, which have less plant matter, or incognito, which are more liquid extracts, would have less interference in the whirlpool. We haven't necessarily tried the liquid extracts yet, but with like cryo, it seemed like it was having a similar effect on reducing the thiol output. So we still don't really know what's going on there, why that's happening. But heavy whirlpool additions definitely seem to reduce the thiol output. So if your goal is maximizing thiols, minimize your whirlpool. Dry hop additions don't seem to actually reduce the thiols, even though I have that up there. But it does reduce the perception of thiol. So we can measure the same levels, but it doesn't come across, if you're heavy dry hopping, they're not nearly as perceivable. They start to get masked by all the other aromas that are coming from hops. So just generally keep hops down if your goal is to maximize thiols. Again, the majority of the precursors come from barley malt. So, minimize the amount of oats and wheat you're using if your goal is to maximize thiol output. And, play with different base malts. I can't point you to any particular, like we know is a super robust base malt because this data is not really out there. But play around with it, and you might find some that seem to give you more pop than others. We do know higher kiln malts have less, so minimize the amount of, everybody should be minimizing the amount of anything. But
I think you got some controversy with that statement.
a lot of amber ale, anyway, I'll not wade into that. Higher kiln malts, less precursor. And then other sources of thiol precursor like phantasm or grape juice, maybe other fruit sources. We've had some interest from fruit manufacturers to try to see if they have precursors. I mean, we'd expect there to be in certain things like passion fruit, which has tons of free thiol. But maybe there's kind of this inert precursor hiding in some other fruit extracts. So we don't know yet. Still need to look at that.
Have you done any experimentation, mixed experimentations?
Yes, some. And that's actually one of the things we recommend to do if you want to pull down on the amount of thiol. So we do seem to get what looks like a dose dependent effect. So if you, for example, if you went, I don't know if I have that slide, but we've done 50% Heliogazer, 50% British 5, the parent strain, and we get about 50% the thiol output when we have the blend compared to all Heliogazer. So those strains are good for blends because they're so potent on their own that you can cut them with other strains and you still get a lot of output. So we've had people ask us if we're going to do a Thiolized Saison. It's not necessary. Just take 50% Saison and pitch it with one of these strains, and you'll get a similar effect. Because it's still going to be putting out tons of thiols, but now you've got thiol yeast, or Saison yeast working too. But it's also something you can do if you want to cut back on the amount of thiol. Like if you want some, but you don't want it overwhelming, you can essentially do a pro rata. You want 75% less, do 75% of the parent strain. Because the good thing about this is you can use the parent strain, and it should be totally stable blend. Neither strain is going to have an advantage over the other because they're essentially the same thing, except one has the ability to release thiols. So in theory, you could actually use that 75-25 blend for 10 generations, and it's not really going to shift in one direction or the other because neither strain has an advantage over the other. So yeah, if you want to lessen the amount of thiol perception, increase the amount of whirlpool hops you're using, or you can mask it with very high dry hopping rates. So yeah, up the hop intensity and lower the thiol intensity. And then increase the amount of wheat and oats and things like that you're using if you want to cut back on it. Oh, I did have that data. So this is what happens if you blend Heliogazer with the parental British 5 strain. So in this case, it's not quite 50, it's a little more than 50%, but definitely an effect. We actually did many more combinations between these, but this is all I wanted to pay for. So sent 3 samples off. We did sensory on all of them. We could definitely tell, at least until our noses were blown out, that there was a difference depending on the ratio, but the machine confirmed that. So things you might try. again, you know, like Helio Gazer, Cosmic Punch, you can use for any IPAs already. It's going to add a note that's, you know, there to some degree already, but it's going to be much more intense. Cosmic Punch is going to add just a little bit of a nudge, whereas Helio Gazer is going to be much more intense and unavoidable. There are a lot of complex interactions that we don't fully understand. We're testing these things all the time. Again, all these masking things we're seeing where even though we can measure huge amounts, it's not nearly as perceivable. And there's probably going to be dry hop combinations that work better than others. Some of these combinations of aromas just come out kind of weird. So it's going to take some exploration to figure out the ones you like more. If you ever really want to know what the files are like in isolation, then make a blonde ale because that's when there's no masking and all you're getting is files, and that's going to be something you probably have not smelled before. You may not like it, and again, you know, and again, like maybe do like 10% thiolized along with 90% non-thiolized in a blonde to give you. It's going to be noticeable for sure, but not nearly as overwhelming, and that will give you a really good sense of the files in isolation, what aromas you're going to be looking for, and then think about how you might want that aroma added to the other styles. So it's a good way to kind of just explore those aromas in isolation.
What was it, parent Lunar?
British. Oh wait, Lunar Crush. That's our Mexican one. Those are some of the ones that are like blended to reduce the thiol output, but in like a very simple like Mexican lager grist. I like those. But it might be an acquired taste because it's like don't call it a Mexican lager or serve it to anybody calling it a Mexican lager.
Yeah, but if you're gonna make man of lager, it's actually the best yeast to use the Mexican lager.
Yeah,
It really comes out.
I like that.
Is that the only lager you tried before?
No, we first tried a German lager strain for the but we tried to find a lager strain that had at least kind of native sulfur output, because these strains are also putting in more sulfur, so it's not just too far. So that's the reason we chose that.
So the fourth sample we have is with Helio Gazer from Sabbatical. So that's the purple can. Tables.
Yes.
I think.
I have the hop seed.
Okay.
Check. He said Cascade Mash Hop Simcoe and 630. Must be a
HPC 630.
yeah, and dry hop Simcoe Citra 630 may be wrong on dry hop though. So match hop a Cascade.
Caddies. Yeah, I mean, our view on these is you're, you know, again, sustainability argument there to pull out more aroma from the ingredients we already have at hand. You can kind of lower the amount of hops you're using and still get a pretty big aromatic impact. And, you know, hops are very intensive, water intensive, chemical intensive to grow. So it's a way to get a lot of flavor with using less hops. And it gives you a lot of exploration potential to try the regional malts. There's every reason to believe there's going to be some variations in the amount of precursors available depending on where your malt comes from. I wish I could tell you, you know, what that's going to be, but it's going to take some exploration, and I would expect there to be variations depending on your source of malt. And also gives you an opportunity to support, you know, your local maltsters and growers.
Yeah, your local maltsters probably is just that.
I've heard
that.
So we do not know everything. We still continue to learn. There's probably some other thiol precursors that these yeasts are acting on that we'd like to identify. We'd like to figure out which varieties tend to work well. I mean, some of that's subjective, but we definitely had some results that were different and some that were more pleasing than others. And then trying to figure out what's working better than others. continue to try to work on mash hopping. Again, I think, it's something that I would explore some maybe with Cosmic Punch, but probably would not with the newer Pappy drive strains. They don't need any help. And you might be adding things that aren't, that you don't want there. But I would say most of all, they don't need help. There's already lots of precursor available in the malt.
Do you think stressing the yeast like under pitching or under time, over time, gonna do anything different?
It
Differently?
should not affect the enzyme.
It's still gonna be there or not?
Yeah, so, but where it will affect it is higher temperature should volatilize more, so you probably have less in your beer, because more fermented, but more cold might retain more.
Under pitching stressing it?
It might affect other things the yeast is doing, but not.
All good, it's already turned up, already there.
Yeah,
we can
because
take-
the reason that house people that eat.
Yeah, it's already on.
But that's.
So this is what's in the,
What are you
the
gonna do? You get
1 and
this,
4,
this, do this.
the last one with the
Under
cutie
control,
of
pitch, higher, lower, more.
So you add some grape skins to the whirlpool. I think, Brent, I think you got all those when you're, for the 1 and 4, right?
Yeah, that's what it said.
Yeah.
Okay.
What do you?
So Will is the owner.
You guys get mad, it's gonna go 6:30, Whirlpool, press, let's go agree for Whirlpool.
That was it. So this is the crew at our Christmas party last year. In there somewhere. Oh yeah. So this time, so what my evolution of my beer is, so before yesterday I had a big full long beard, I shaved it down to this, and then I shaved this off. So that's what's in there.
It's great.
So
Anyway,
you're the other co-owner is
Mark, well, yeah, he's not acknowledged on here. He's the guy in the back at the top.
like doing this?
Yeah,
Okay.
Yeah, happy to take any other, I'm gonna answer questions along the way, but we have others.
How long have you guys been in business?
9 years.
9 years. And how did you get all your original yeast strains?
Lots of places.
Okay.
I mean, some of them I had, again, we had a huge yeast bank at the club I was in college. Took all those down with me to Texas and brought them back up to
How do you
Illinois.
make sure they're pure?
Huh?
How do you make sure they're pure?
You can, just microbiology, so you can take, yeah, so just streak out so you get a single colony, and then you have a clonal colony.
And how do you make sure that there's not mutations that happen?
I mean, there's going to be, just, yeah. So, I mean, the way we maintain our strains is we have a stock freezer,
Sure.
and we start from there every time crop. It's gonna be mutating along the path.
Yeah.
It just happens every time.
So what market share do you guys have?
That's true. Don't know. I don't know. Of the liquid yeast manufacturers, my guess is we're their second. White
Yeah.
Then either us.
How does wine yeast fit into that?
They were probably the second biggest like 10 years ago. And I think they were
I mean, I knew.
Now,
You said a lot.
Yeast, now there's like an asexual, budding. How do you promote like hybridization or crossing of
starve them essentially.
Okay.
And then they'll sporulate. And then when they do that, they go into haplorous, a single copy from some state. And when they go vegetative again, they see a cell in opposite J type. Do the thing.
Sure. So when you see people trying to recreate a yeast strain and then eventually freeze it and save it, what does that impact have on freezing and then bringing it back in?
It kills, it kills a portion of the population. Everything freezes, kills a portion.
And a starter would bring half back?
No, then they'll eat everything available, so they'll produce as many new cells as the medium will support. So it doesn't, it doesn't matter if you start with 5 cells or 100 cells, you generate pretty much the same amount of cells.
And is that basically the same with like the starter? So like you take one package. You're doing it.
Yeah, I mean, there's not, so in a starter, there wouldn't be anything. Putting 2 packs or 1 pack in 1 liter order will essentially generate the same amount.
But if you do one pack, it's like 4,000. So you're like the big, you're 5.
Yeah, I would still only do 1 pack.
So.
It'll grow into it. I mean, there's a limit, so you, I mean, so you can't take that, and say I can.
Yeah.
It does break down eventually, that's because they're only getting you can have pure, it's gonna get affected. Even though 1 pack.
So you got a question from Wilson. So, says, will you have anything similar to what's overlap there? I think it's similar to like Philly sour.
Yeah.
And where can you get one of those?
Wait, he's a customer, right?
Yeah, yeah. He'll send you one. So, no, Frank, okay, we have a question. So Will from Sabbatical asked about the lactic yeast, which is a lactic acid producing yeast, like you've heard of Lala Philly sour. So he asked if Omega is looking at doing something similar to that.
So we have made some. The problem we're having right now is getting it to make a reasonable amount. So they're making way too much. So that
Too much acid?
yes. So the one we've made, so if we start with like a 12 Plato, where it'll attenuate to like 50% and then kill itself, and it's at a pH of like 2.5. So
Is that a genetic modified yeast?
yeah, yeah. So that's taking. We actually took the LDH enzyme, so that's the enzyme that can take pyruvate, turn it into lactic acid. From our lactobacillus that we sell. So it's that gene inside yeast. We have what we think is a novel use for it in making NA sours. So if you do a very, very low gravity work and use it there, you get a high amount of acid produced, but there's just not that much fuel for it. And it turns like half of the sugar into acid and half of it into alcohol. So you end up with a low ABV, nice, and 0.5% alcohol, tart, flavorful. So it might have that niche, but we can't even grow it to use for.
It just stops reproducing.
Yeah, it kills itself.
So in your research and development, like, what are the new strains that you guys are feeling is going to be the next big thing? Like, how did you come upon like, what's the next thing?
We've been doing a ton of research on haze, and because there's very much strain dependence in stable haze, so for like any haze, trying to figure out the genetic component of the strains that make with stable haze. So like Chico, for example, start where it doesn't have that. It's hard to make it. So we discovered that Chico have a version of Chico that's good.
You can wear that shirt. You should.
So I think.
It's a
So you
good
finding
one.
that the craft beer industry
So as far as we can tell from what we make, I would say it's 30, probably a third of the yeast we produce is used in everything.
So when you first started, were you more, did you see more brewers starting out with you, or after brewing right away off the bat? We're in on this, and we want to see what we can.
It's been pro from bread and butter. It's probably like 90%.
Okay. Brought it home.
Serious.
What's the furthest you've sent yeast, like distance?
New Zealand or Japan?
So Lance came all the way from Chicago, so if we don't have any other questions for him, we want to get him out of here because he's got to drive home,
Yep.
so.
Good job, man.